Hello viewers, welcome to my channel. Today we will discuss about the microbiology question paper of GATE XL 2020. Some brief notes regarding the question and their related options are also provided here, which will be beneficial for you. So stay tuned in this video. The first question. The technique of microbial pure culture was pioneered by option A. Edward Jenner, option B. Louis Pasteur, option C. Robert Hooke, and option D. Robert Koch. The correct answer is option D. Robert Koch. Now, what do you mean by pure culture? A pure culture is a culture containing only one type of microorganism. Koch developed the culture media using meat extracts and protein digests because of their similarity to body fluids. Robert Koch also discovered Mycobacterium tuberculosis, the causative agent of tuberculosis, Vibrio cholerae, the causative organism of cholera, Bacillus anthracis, the causative organism of anthrax. Edward Jenner was an English physician and scientist who pioneered the concept of vaccines and created the smallpox vaccine, the world's first vaccine. So, world first vaccine is smallpox vaccine that was developed by Edward Jenner. Louis Pasteur was a French chemist and microbiologist, renowned for his discoveries like microbial fermentation, rabies vaccine, and pasteurization, the last of which was named after him. Last is Robert Hooke. He was an English scientist who discovered cell, the building block of all life. Next question. The antibacterial trimethoprim is an inhibitor of option A. Dihydrofolate reductase, option B. Dihydropteroate synthetase, option C. N5N10 methynyl tetrahydrofolate synthetase, and option D is serine hydroxymethyl transferase. And the correct answer is option A. Dihydrofolate reductase. Trimethoprim binds to dihydrofolate reductase and inhibits the reduction of dihydrofolic acid, that is DHF, to tetrahydrofolic acid, that is THF. Sulfamethoxazole inhibits dihydropteroate synthetase. It inhibits the conversion of PABA, that is para-aminobenzoic acid, to dihydrofolic acid. Next question. Choose the correct taxonomical hierarchy among the following. Here, different orders of the taxonomical hierarchy is mentioned and you have to choose the correct one. First option is species, followed by genus, followed by family, followed by order, followed by class, followed by phylum, followed by domain. Option B is species, genus, order, class, family, phylum, domain. Option C is species, followed by genus, followed by order, followed by family, class, phylum, domain. And option D is species, genus, family, class, order, phylum, domain. And the correct answer is option 1. That is species, followed by genus, followed by family, followed by order, followed by class, followed by phylum, followed by domain. So, what do we mean by taxonomic hierarchy? Taxonomic hierarchy refers to the sequence of categories in increasing or decreasing order. Kingdom is the highest rank and species is the lowest rank in the hierarchy. Next question. Shifting a Saccharomyces cerevisiae culture from fermentative to aerobic respiratory mode will option A. Decrease carbon dioxide production, option B. Increase alcohol production, option C. Increase glucose consumption, and option D is decrease ATP generation per mole of glucose. So, the general sequence of reaction, that is glucose to pyruvate, is common. Sorry, this glucose to pyruvate or pyruvates. In fermentation, it is converted to first to acetaldehyde, then to ethanol. Whereas in aerobic respiration, the pyruvate first is converted to acetyl CoA, and then it goes to the TCA cycle or Krebs cycles. So during aerobic respiration, ATP production is higher as compared to fermentation, and glucose consumption and carbon dioxide production decreased as compared to fermentation. So the correct answer will be option A, decrease carbon dioxide production. So when Saccharomyces cerevisiae culture is shifting from fermentative to aerobic respiratory mode, then the carbon dioxide production will be decreased, whereas ATP production will be increased. Next question. Which one of the following disease is treated by neuraminidase inhibitor? Option A, chickenpox. Option B, polio. Option C, influenza. And option D is Japanese encephalitis. And the correct answer is influenza. The neuraminidase inhibitor are a class of drug which block neuraminidase enzyme. They are a commonly used antiviral drug type against the influenza. The viral neuraminidase are essential for influenza reproduction, facilitating viral budding from the host cell. Oseltamivir, zanamivir, linanivir, peramivir belong to this class. Unlike the M2 inhibitors, which work only against influenza A virus, neuraminidase acts against both influenza A and influenza B. Examples of influenza A are H1N1, H3N2, etc. So H for hemagglutinin and N for neuraminidase, which is essential for this viral influenza virus reproduction. Next question. Which of the following does not provide three-dimensional images? Option A, atomic force microscopy. Option B, confocal scanning laser microscopy. Option C, differential interface contrast microscopy. Option D, phase contrast microscopy. And the correct answer is phase contrast microscopy. Other three options, that is atomic force microscopy, confocal scanning laser microscopy, differential interface contrast microscopy, all provide three-dimensional image, but not the phase contrast. It provides two-dimensional image. AFM, that is atomic force microscopy, can be used to study the structure and mechanical properties of protein complexes and assemblies. For example, it is used to image microtubules and measure their stiffness. AFM can be used to attempt to distinguish cancer cells and normal cells based on the hardness of the cells, because cancer cells are hard as they are highly differentiated cell. So cancer cells is hard than the normal cell. This AFM is also used to evaluate the interaction between a specific cell and its neighboring cells in a competitive culture system. Next one is the phase contrast microscope, that is used to study the living cells and how they proliferate through cell division. Phase contrast microscope is one of the few methods available to quantify cellular structure and components that does not use fluorescence. Clinically, confocal scanning laser microscopy is used in the evaluation of various eye disease and is particularly useful for imaging, qualitative analysis and quantification of endothelial cells of the cornea. This confocal scanning laser microscopy is used for localizing and identifying the presence of filamentary fungal elements in the corneal stroma in cases of keratomycosis. enabling rapid diagnosis and thereby early institution of differentiative therapy. Next one is the differential interface contrast microscopy. It is also known as Nomarsky interface contrast or Nomarsky microscopy. It is used to enhance the contrast in unstained transparent samples. The image has the appearance of three-dimensional object under very oblique illumination. It is used for imaging live and unstained biological samples, such as smear from tissue culture and individual waterborne single-celled organisms. Its resolutions and clarity in conditions such as this are unrivaled among standard optical microscopy techniques. One non-biological area where the differential interface contrast microscopy is used is the analysis of planar silicon semiconductor processing. The image quality when used under suitable condition is outstanding in resolution and almost entirely free of artifacts, unlike the phase contrast microscope. Next question. Which one of the following will increase the resolution of a light microscope? Option A, decreasing the numerical aperture of the objective lens. Option B, using an objective lens with a higher working distance. Option C, using a medium of higher refractive index. Option D, increasing the wavelength of light. And the correct answer is using a medium of higher refractive index. As we know that the D is equal to lambda by 2Na, where lambda is the wavelength of light, Na is the numerical aperture, and Na is equal to small n sin theta, where n is the refractive index of the medium in which lens works, and theta is half angle of the cone of light entering the objective. D less, resolution high. So, using a medium of higher refractive index, that is if the n is higher, then Na become higher. As a result, D become less. So, resolution become high. But in case of increasing the wavelength of light, the D become higher. So, resolution will be less as the D is high. So, this option is not correct. And decreasing the numerical aperture. If the numerical aperture, that is Na, is decreased, D will be high. So, resolution will be less. See, this option is also not correct. But in case of this C option, that is using a medium with higher refractive index, that is Na is equal to small n sin theta. So, Na become higher. As a result, D become less. So, resolution become high. Next question. Which of the following condition favors maximum expression of lac operon genes in E. coli? Option A, glucose low, lactose low, CAMP high. Option B, glucose high, lactose low, cyclic AMP high. Option C, glucose low, lactose high and cyclic AMP high. Option D, glucose high, lactose high, cyclic AMP low. And the correct answer is option C, that is glucose low, lactose high, cyclic AMP level high. Then the expression of lac operon genes in E. coli will be maximum. So when glucose level is low, CAP, that is catabolite activated protein, and cyclic AMP bind to the CAP site, which favors maximum expression of the lac operon. When lactose high, lactose bind to the lac repressor. The lac repressor becomes inactive so that it is unable to bind to the operator site. So lac operon is transcribed at a maximum rate. Next question. Match the following cellular organelle in group 1 with its function in group 2. In group 1, the cellular organelles are Golgi apparatus, Q is nucleolus, R is peroxisome, and S is proteasome. In group 2, the functions of the cellular organelles are: 1 is lipid degradation, option 2 is protein degradation, Option 3, protein shorting. Option 4 is ribosomal RNA synthesis. So, nucleolus helps in the ribosomal RNA synthesis, whereas this proteasome helps in protein degradation. So, Q is R and S is 2. Q is R, S is 2. Q is R, S is 2. It is option B. Not in another option, Q is R and S is 2. So, let's see another option. Golgi apparatus. It helps in protein shorting. So, P is 3 and the rest is peroxisome. So, it helps in lipid degradation. So, the correct answer will be option B, that is P3, that is Golgi apparatus, that helps in protein shorting. QR, that is nucleolus, that helps in ribosomal RNA synthesis. Peroxisome, that helps in lipid degradation. And proteasome, that helps in protein degradation. Next question. It is one mathematical problem. 250 microliter of bacteriophage stock containing 8 into 10 to the power 8 phage per ml is added to 500 microliter of E. coli culture containing 4 into 10 to the power 8 cells per ml. The multiplicity of infection is, so MOI, that is multiplicity of infection, is equal to phage divided by cells. So we have to calculate the total number of phage and total number of E. coli cells. So number of bacteriophage present in 250 microliter of stock is 2 into 10 to the power 8, and number of E. coli cells present in 500 microliter of culture is like that, 2 into 10 to the power 8. So the multiplicity of infection will be phages by cell, that is 2 into 10 to the power 8 divided by 2 into 10 to the power 8, that is 1. So the multiplicity of infection will be 1. The MOI related mathematical problem is already discussed in one of my previous video. Link is shared in the description box. You can check it. Next question. Digestion of an immunoglobulin molecule with pepsin will not. Option 1. Option A is generate a bivalent antigen binding fragment. Option B, generate monovalent antigen binding fragments. Option C, destroy the complement binding site. Now, option D, cleave the heavy chain of IgG molecule. Here you have to choose the incorrect option, because in the question it is mentioned that digestion of immunoglobulin G molecule with pepsin will not. So you have to choose the incorrect option, and the correct option and the answer will be B, generate monovalent antigen binding fragments. So pepsin is a non-specific endopeptidase. It is active only at acid pH. It is irreversible denatured at neutral or alkaline pH. Pepsin cleave or digest below the disulfide bond of the hinge region. Digestion by enzyme pepsin normally produces one Fab2 fragment and numerous small peptides of Fc. So one Fab2 fragment is bivalent. So the incorrect option is the generate monovalent antigen binding fragment. Here the diagram of the antibody is shown, and cleavage by pepsin papain is also shown, and mercaptoethanol. By digestion with pepsin, it is we have already mentioned that pepsin digests just below the disulfide bond of the hinge region. This is the hinge region, and this is Fc region is the complement binding site, and these two are antigen binding fragment. So digestion with pepsin results in one Fab2 fragment that has two antigen binding site, and the pepsin and the FC regions is fragmented. So the complete binding site is not present. But in case of papain digestion, the papain cleave just above the disulfide bond of the hinge region here. So it creates two Fab fragment. And so if we go to there, so this option is like that: generate Fab2 fragment, that is two bivalent antigen binding fragment. In the image you can we have already discussed, destroy the complete binding site as the FC regions is cleaved in very FC portion is fragmented into small peptides, and cleave the heavy chain of the IgG molecule as it cleave because it cleaves the IgG molecule, it's also mentioned, and generate monovalent antigen binding fragment. It is the monovalent antigen binding fragment is generated by the papain. Okay. So now next question. Match the process involved in nitrogen and sulfur cycle in group 1 with the corresponding microbe in group 2. Here in group 1, the nitrogen and sulfur cycle process is mentioned. P is denitrification, Q is nitrogen fixation by free living microbes, R is oxidation of H2S to sulfur, and S is nitrogen fixation by a symbiotic microbe. In group 2, option 1 is Azotobacter, 2 is Vegetua, 3 is Pseudomonas, and 4 is Rhizobium. So most common is, we will know that nitrogen fixation by a symbiotic microbe is Rhizobium. So S is 4. In option B and option D, both option S is 4 mentioned. Now let's discuss about another option. Denitrification. Denitrification is done by Pseudomonas. So it is P is 3. So P is 3 in option D. Now another option is Q, that is nitrogen fixation by free living microbe is Azotobacter. That is Q is 1. So in option B is Q is 1, and last is R is 2. R is oxidation denitrification. P is 3. P is 3, and oxidation of sulfur to H2S to sulfur, that is R is 2 and P is 3. So the correct answer will be option D, that is denitrification by Pseudomonas, nitrogen fixation free living microbe is Q is 1, nitrogen fixation symbiotic microbe is S is 4, and R is 2. Vegetua, oxidation of H2S to sulfur. So denitrification by Pseudomonas, nitrogen fixation by free living microbe is Azotobacter, oxidation of H2S to sulfur is Vegetua, nitrogen fixation by symbiotic microbe is Rhizobium. Next question. Determine the correct or otherwise the following assertion and the reason. Assertion is mentioned as A and reason is R. Assertion: diphtheria exotoxin is an example of AB type toxin. It is correct because diphtheria toxin is an AB toxin. Diphtheria toxin is an exotoxin and it is AB type of toxin. Next is A component of the toxin is released from the host cells, while the B component inhibit protein synthesis and kill the host cells. No, this the reason is it is not correct, because the B subunit is helps in binding. So in option A is both A and R is true. No, because A is correct but R is not true. Another thing you have to we don't have to check. B is both A and R is true. It is also not correct. Both A and R is false. Option C, no, A is correct and B is false. Now option D is A is true but A is true but R is false. It is correct. So option D is the answer will be option D. That is A is true but R is false. So diphtheria toxin is an AB type toxin. It is an exotoxin. A component is responsible for the catalytic activity and the toxin, that is the toxic action, and the B subunit is responsible for the binding capacity. Next question. An example of a differential and selective medium in which colonies of gram-negative bacteria produce large amounts of acidic products and appear green with a metallic sheen is option A, blood agar, option B, EMB agar, option C, McConkey agar, and option D is mannitol salt agar. And the correct option is option B, that is EMB agar, that is eosin methylene blue agar. Two dyes, eosin Y and methylene blue, inhibit the growth of gram-positive bacteria. They also react with the acidic products released by certain gram-negative bacteria when they use lactose or sucrose as a carbon and energy source. Colonies of gram-negative bacteria that produce large amounts of acidic products have a green metallic sheen. For example, the fecal bacteria E. coli. So in my previous video, I have already discussed about the colony characteristics growing on different culture media. So you can check it. The link will be shared in the description box. Next question. Which one of the following is an example of substrate level phosphorylation? Option A is glucose to glucose 6-phosphate. Option B, fructose 6-phosphate to fructose 1,6-bisphosphate. Option C is 1,3-bisphosphoglycerate to 3-phosphoglycerate. Option D is 2-phosphoglycerate to phosphoenol pyruvate. And the correct answer is 1,3-bisphosphoglycerate to 3-phosphoglycerate. Now, what is substrate level phosphorylation? Substrate level phosphorylation is a metabolism reaction that results in the production of ATP or GTP by the transfer of a phosphate group from a substrate directly to ADP or GTP. 1,3-bisphosphoglycerate dephosphorylated via phosphoglycerate kinase and producing 3-phosphoglycerate and ATP through a substrate level phosphorylation. Next, it is also a mathematical problem. A bacterial culture containing 3 × 10⁵ live cells was exposed to a newly developed sterilizing agent. After 30 minutes of exposure, 3 live cells remained in the culture. The decimal reduction time in minutes for a new agent is, so the decimal reduction time D is equal to T2 minus T1 divided by log N0 by N, where the N0 is the initial bacterial population, N is the surviving bacterial population, T1 is the initial time, and T2 is the final time. Here, the final time is T2 is equal to 30 minutes. And T1, that is initial time, is 0 minute. N0, that is the initial bacterial population, here is 3 × 10⁵. Whereas the surviving bacterial population is 3. Here, as it is mentioned, the 3 live cells remained in the culture. So the surviving cells is 3. By putting this value, the answer will be 6 minutes. Next question. It is also a mathematical problem. A continuous culture carried out in a chemostat is said to flow rate of 40 ml per hour. The culture volume is equivalent to that of cubical container having 10 centimeter side. The dilution rate of this system is, so dilution rate is equal to flow rate divided by the volume of the culture. Here it is mentioned that the culture volume is equivalent to the cubical container having 10 sides. So the volume of 10 sides cubes is 10 semi-cube. So that is 1/3 is a semi-cube. As you know that 1 semi-cube is equal to 1 ml. So the volume is equal to 1000 ml. Here also mentioned that the flow rate of the chemostat culture is 40 ml per hour. By putting this value, D is equal to 40 ml per hour divided by 1000 ml. That is 0.04 hour inverge. So the answer will be 0.04 hour inverge. Thank you. Please like, share and subscribe to this channel.